All right, so everyone, welcome to our last guest speaker presentation for this semester. Starting next week will be our first student presentation. So we'll be making that transition. Our co-host today is Gabriella. I'm not going to try to pronounce her last name, even though she gave me the pronunciation here. Um, Gabriella is a senior here at Andrews. She's from Spokane, Washington. She is a double major, chemistry and math studies in the pre-med program. Her goal is to become an oncologist, but she also does enjoy um, some artistic endeavors such as painting. So Gabriella will go ahead and introduce our speaker for today.
Thank you, Dr. Murray. So our speaker today is Jared C. Lewis. He was born and raised in Effingham, Illinois. He attained his BS in chemistry from the University of Illinois, earned his PhD in chemistry from the University of California, Berkeley, and conducted postdoctoral studies at Caltech. He started his independent career at the University of Chicago in 2011, and in 2018, he moved to the Indiana University, where he currently acts as a professor of chemistry. Please join me in welcoming our speaker for today.
All right. Thanks so much, Gabriella, for the introduction and Desmond for the invitation to come and speak. I love doing this. We do it all the time at IU. I give a lecture similar to this to our undergraduates. I will say I completely retooled the whole thing this morning, so we'll see how that goes. That's part of why I don't normally time myself, but I am definitely keeping an eye on the timer right now. And you know, the goal here is to give you a flavor of the type of research my group does, but you know, obviously from the standpoint of um, uh, a more introduction, you know, to the area. And I'll show you some of what we do, but after a pretty good deal of introduction. Okay. So um, where I like to start with this is, you know, my interest is in synthesis. I like to make things. Okay. And chemical synthesis is required for making pharmaceuticals, agrochemicals, fine chemicals, plastics, all these materials that you find around you. Okay. So that's critical. And fundamentally, this involves manipulating molecules. And I think, you know, to make sure everybody's on the same page, right? Like it's always useful to give a metaphor here. And a lot of chemists will go back to Legos, right? Blocks of some kind. And everybody's familiar with this series of instructions, right? You go through and you build complexity by snapping the blocks together in a particular order, and you get to a robot, right? And or whatever, right? And the critical thing that's not shown in those instructions, but that you all implicitly know, of course, is that you are involved, right? You're part of that manipulation of matter to allow a particular assembly to occur. It's not as if you just take all the blocks in the bags, shake them up for a bit, and out comes the robot, right? You are required to follow those instructions. Your hands are required to put those blocks together. Now, when we compare this to chemical synthesis, it's obviously pretty different. Now, on one hand, you can draw up a scheme that looks just like the Lego instructions you grew up with, right? Um, there are some differences, obviously. We're relying on the chemoselectivity of reagents to interact with different functional groups. Everybody's learned about how to make an acid chloride or how to hydrogenate a double bond or how to, you know, alkylate an amine, right? We know those reactivities. But the critical thing that's missing here, of course, is your hand is not, in fact, involved, right? You can't manipulate matter at that level. Again, we rely on chemoselectivity to dictate these reactions. There are other differences, of course. There are byproducts and protecting groups and things like that in synthesis that certainly don't exist in Legos. But the bigger issue here is how you manipulate matter is fundamentally different, right? Now, that sort of brings in one of the primary interests of my entire academic career, starting when I was a graduate student, and that's catalysis. And I'll bring these two areas together, what I was talking about with manipulation of matter and catalysis here in a moment. But we need to be on the same page with respect to what we're talking about, right? So a catalyst is some species that lowers a reaction barrier, without being consumed. And it typically does so via a unique reaction mechanism, right? So let's say we have some starting materials and some products, right, on one side of a reaction and on another. And I mentioned hydrogenation in that first slide. We're going to kind of use that as a basic, exemplar reaction throughout, right? So H2 in an olefin going to ethane, let's say. Now, uncatalyzed reaction, if you could do that, it's going to be some high barrier with like one, a transition state. What we mean by this unique reaction mechanism is that you might have an alternate pathway whereby you maybe break that process up into a series of steps, and those series of steps have a lower overall barrier than the uncatalyzed pathway. So that's what we're talking about here, right? There are lots of different kinds of catalysts out there. You can have simple acid-base catalysis, you can have metals, you can have organocatalysis. We're going to be talking about enzymes, of course, but there's a lot out there that can do this. So, for example, here's maybe taking this sort of energy scheme and bringing it to a little bit more chemical, space. You could have metal surfaces that catalyze reactions. You're all familiar with catalytic converters, right? Those are metal surfaces that catalyze, different decomposition of different, exhaust gases. But you can also catalyze hydrogenation on surfaces like palladium or platinum, and maybe you've done this in a synthesis lab where ethene comes in and it binds to the surface. And that could be, say, this initial step, right? We have a step where we form a bound intermediate. And then hydrogen is going to do that too, and it gets split. And then hydrogen can add to the adsorbed ethylene, right? Maybe that's another of these little wells and barriers. And then finally, you release your alkane product, right? You can do this in solution. So instead of having a surface, you can have metal complexes. This is another popular lab that students do often in sort of early organic courses, where we have hydrogen adding to a metal complex and ultimately to an olefin to spit out the alkane product, just as we saw on a surface. And of course, if you put chiral ligands on your rhodium complex, remember your hands are chiral, we have these opposite senses of chirality. That's very important because biological systems are chiral. So if we put chiral ligands on our metal complex, we can make enantioselective catalysis work. We make single enantiomers of the products. And that sort of thing, selective catalysis, can greatly improve efficiency. So you see, catalysts can take a lot of different forms. But one thing I hope you can take away from this is all these do basically the same thing. They selectively bind a transition state relative to a reactant state. What does that mean? They're binding a low energy intermediate tighter than they bind the transition state for that intermediate. Or sorry, they bind the transition state tighter than the reactant state. I got that backwards, sorry. Bind the transition state tighter than the reactant state. If you do that, you lower the barrier, you make the reaction go faster. Schematically, you can see it this way. We have a substrate, we have a catalyst. We see a little bit of interaction between the catalyst and the substrate. That's what the dashed lines are showing. But a lot of interaction with the transition state. So you're pulling that transition state energy down and then, making the reaction more favorable. You must have some release of product then also. Now, importantly, this is where this comes back to that first slide. That means that the catalyst is your way of manipulating matter at a level that you have the same control where you did with building blocks, right? You're able to interact with a transition state directly, which I think is just a profound, idea, okay? Now, I'm going to talk about enzymes. So, enzymes are protein-based catalysts that evolved in nature, all right? They catalyze nearly all metabolic processes. Maybe you've seen one of these gigantic metabolic network maps, on a biochemistry wall. All I'm meaning to show here is you got a bunch of molecules, connected by arrows, and over all those arrows is some enzyme, okay? So that's all I'm meaning to illustrate. And you have these very complex networks that are going on in your body all the time, right? Now, what's interesting, if I can bring that back to synthetic chemistry, is that often enzymes will work on synthetic substrates, not just the ones that they evolved to catalyze, right? And if that's true, now we can start to think about biocatalysis. How can we use enzymes that evolved in nature to do stuff, make reactions that we care about, to catalyze synthetic reactions? All right. So if we want to think about that, we need to understand a little bit about enzyme activity. And there are a few basic features that most enzymes adhere to. One is called the fold. So this is the three-dimensional shape of an enzyme. We're going to show some examples in a minute. One is binding. I just said on the previous slide that catalysis, binding is central to catalysis, right? You want to bind that transition state tighter than the reactant state. And if you do that, you can make the reaction go faster. And that involves interactions between the enzymes, substrates, transition states, products, all these things. Okay. There's, of course, chemistry involved, right? We have the reactivity of amino acid side chains and cofactors that are all dictating the reactivity of the enzyme. And there are dynamics, okay? That's the movement of residues within an enzyme that allow for substrate to come in and exit. It's not universal. It's more important in some enzymes than others, so I'm not going to emphasize it, but it's worth throwing out there, okay? So your enzyme activity depends on these things. I'm going to give you a bit of an introduction with an enzyme called Old Yellow enzyme. And I'm taking all these figures from this review. So if you're interested in this, you can read more about it here. I thought it was great because it really nicely illustrated these very simple ideas, at least the first three very clearly. And that's what I wanted to focus on. So this enzyme, Old Yellow enzyme, catalyzes alkene hydrogenation. So I obviously also chose it because my previous couple slides ago emphasized alkene hydrogenation from a synthetic perspective. So now I'm showing you how it happens in nature. Okay, so this enzyme uses a flavin cofactor and NADPH as a reducing agent, and it adds H2 formally across this double bond to give you the alkane product. Okay, now you do have to have an electron withdrawing group, so that's a requirement for the enzyme. But again, the way it works is it's going to add a hydride equivalent to the electron deficient side of the olefin, and that'll generate, say, an enolate intermediate, and then that gets protonated. So you're getting your H2 from proton and hydride sources, okay? It can work on these types of substrates shown down here. So fairly different. You have cyclic enones, you have acyclic enones, different branch patterns around the olefin. And you can access products that are very important. So you've all encountered ibuprofen, I presume, and you can access that with an ene reductase. Here's a neurokinin 1 antagonist precursor. Here's a simple building block. All these things you can access with these old yellow enzyme and other ene reductases, they're called. So quite a useful enzyme class. So now, what about those features I told you about? Well, one I told you was the fold. If you have ever seen, an enzyme before, you've seen these sorts of ribbon diagrams typically representing them, okay? And this is the structure, from the crystal structure of old yellow enzyme, okay? In D is just another ene reductase. And so what this is meant to show is the fold is quite distinct. I think even if you don't know anything about enzymes, you can see some things are different between these structures. If you squint, you can see that a lot of it is actually the same, that you have an alpha beta barrel. So you have this beta, these flat sheets in the middle are beta sheets. And then these helices on the outside are the helices that comprise the barrel. But you can see that in old yellow enzyme versus this one, you see these loops in this enzyme are just simple lines, right? They're really simple loops connecting different sheets and helices. But in old yellow enzyme, they're a little fancier, right? They have some helical behavior, things like that. So those differences in the fold give rise to differences in catalysis, in binding. That happens because they cluster amino acids in the active site differently, all right? So you can see all these different amino acids in this little constellation around the active site. These are flavin enzymes. So the flavin cofactor is shown here. You see it says FMN, and there's the gray flavin. What this molecule sitting on top is, it's just a benzaldehyde, paramethoxybenzaldehyde, I guess para-anisaldehyde. And that's an inhibitor for these enzymes, actually. So that's not a substrate, but it's showing you where the substrate binds. And look at this. Both these enzymes, A and D, have the same inhibitor bound. But look, the aldehyde in D is pointing the other direction. That constellation of amino acids is slightly different, so it binds the substrate slightly differently. which gives rise to differences in catalysis when it actually binds an enone substrate. What about that last bit? Well, the chemistry is, of course, critical here. So we get to zoom in a little bit more on just old yellow enzyme. There's the FMN cofactor. Now we're seeing an actual substrate bound, right? So you see here's an enone, and that's the model of it shown. And it can bind in two different orientations is what they're showing here. But the chemistry is similar in both cases, right? We have hydrogen bonding to activate the carbonyl. Remember I told you that the Flavin cofactor donates a hydride. Okay, so there's the hydride attacking the enone. And then when you generate that enolate, you have a proton right here to protonate it, to add hydrogen, okay, across that double bond. So that's the chemistry that we're talking about. Now, if you think about it a little more, and we don't have time to go into it, lots of interesting features from a binding perspective. The stereochemistry matters, right? We're generating a single enantiomer of the product based on the face of the enone that you add the hydride and the proton to. The dynamics are involved in getting the substrate into that active site and the product out, but we're not going to talk a lot about that today, okay? So I think that's a really nice introduction, to how enzymes work from that simple enzyme class and how it relates to more conventional reactions like hydrogenation and to catalysis in general that you might have seen in say, an introductory organic class. So if I could recap a little bit some of the benefits or some of the features of enzyme catalysis, I didn't emphasize it, but these things are genetically encoded and microbially produced. So that means that you can order the gene encoding an enzyme, put it in E. coli, and the E. coli will make it for you. So you don't have to make your rhodium catalyst or anything anymore. We just get the DNA, E. coli will make it for us. Enzymes are very highly active. They have very high selectivity and very mild reaction conditions. They have to because they originally evolved to do this stuff in a living organism. You can't heat it up to 100 degrees C or things like that. That's not quite true. In some cases, there are deep sea, thermal vents where organisms do in fact live and can survive. But your average, terrestrial organism or bacterium doesn't have that capability. So they're very mild reaction conditions. You put all this together, you can control reactive intermediates, override substrate reactivity, and that's something that chemists very much want. It gets back to that control of matter again. So exactly what I was saying at the start, I like to think of enzymes as allowing us to manipulate a substrate, to manipulate the chemical steps in that ene reduction to enable selective reaction, right? But if you're thinking through this, remember that's all coming from this constellation of residues that are sort of precisely placed around the substrate that enable that selectivity and activity. And that's all due to the fold, right, that I told you about, the fold that orients those residues in just the right way. And so if you're thinking about this, you're like, is that general at all? What if I don't want to work on that substrate? Are all those interactions going to be just right for my substrate? And so there's another concept you need to be thinking of, and that's generalizability. All right. You don't necessarily want a catalyst that works on everything, right? Because if it works on everything, it's not selective. The rates aren't going to be high, that sort of stuff. The selectivity can't possibly be high. What you want is a catalyst that you can tune to do whatever it is you want rapidly. All right. So how do we do this? Well, it turns out there's a tool called directed evolution that is used to tune enzyme reactivity and selectivity. So very briefly, let's say we have some enzyme that can take this sort of made up substrate. I just drew the structure, and it can stick an R group on it. Maybe the R group is a halogen. Maybe it's a hydroxyl, who knows, right? But we're interested in that activity. We want to be able to stick an R group on our compound, but maybe we want to stick it somewhere else, put it on somewhere else in the molecule. Well, what we can do is take the gene, this is representing a plasmid encoding a gene, little arrows represent genes, take the gene encoding that enzyme, and we can diversify it. And there are lots of tools that we can use to make copies of our gene that have mutations in it. And when we express the gene products, the enzymes themselves, in 96 well plates, where each well in our 96 well plate has a different enzyme variant, now we can screen those variants for activity. And perhaps we have assays that will allow us to look at enzymes that stick the R group in different places. And if we like that product, let's say that's $1 billion drug and we want to make that highly selectively, well, we take the gene encoding that enzyme, the one that gave us that product, and repeat this process over and over and over until we get only that product in very high yield, very high selectivity, and whatever other parameters we need to make that on large scale for production of the chemical worldwide. Okay, so we can do that. And it turns out that this is a very important discovery. It was, in fact, the subject of the 2018 Nobel Prize in Chemistry. So Francis Arnold was the winner of this for the directed evolution of enzymes. And these guys did some related stuff with phage display. I'm going to show another picture of Frances because she was my postdoctoral mentor. This is her picking up her medal in Stockholm. So just an amazing story, I think, of this whole science, right? And I want to touch on that a little bit more. So a tiny bit more on directed evolution. So this is just another way of showing that scheme that I showed where you have a gene, you make copies, you put them back into the plasmid, you put them into E. coli, you pick colonies, put them into 96 well plates, and screen for the activity you want and repeat. So the original thing that Francis did in 1993 involved introducing random mutations throughout a gene and then screening to find improved function. Okay, I'm only going to focus on her example right now. What this is, she was looking at a lipase. A lipase cleaves esters. All right, and normally enzymes work in aqueous solution. And what you're seeing here is different concentrations of DMF. And you can see the wild type enzyme. And the number I want you to, this is its activity here. The number I want you to look at is called Kcat over Km. This is how enzymologists measure activity. And you can see that the Kcat over Km for the wild type enzyme is 38 times 10 to the minus 3, okay, per molar per second. That doesn't need to mean anything to you, okay? What does need to matter is the comparison. As we go to 20% DMF, you see that falls off by an order of magnitude to 1.4. And 40% DMF, you lose another order of magnitude. So enzymes don't typically like organic solvent. But what Frances did was had her students introduce mutations into the enzyme and screen those enzymes for improved activity with more DMF around in solution. So you can see the activity of the enzyme goes up, but you see it also, it's kind of going back and forth in 0% DMF because they were screening in increased DMF concentration. And you see that the activity in increased DMF concentration is going up and up till ultimately you get roughly the same order of magnitude activity in 40% DMF that you had in 0% DMF. Okay. Now, her good friend Pam Stimmer did a very similar experiment in 1994 for antibiotic selection. He unfortunately passed away before the Nobel Prize was awarded. Otherwise, he absolutely would have shared the prize with Francis for his discovery or his work the year following. The first example in sort of normal, like enantioselective catalysis was Manfred Rietz, who several years later showed that you could use an enzyme to hydrolyze a lipase to hydrolyze an ester. And you can do so to get, one enantiomer of this product and the other enantiomer of the starting material just left alone. And he's showing that you can gradually increase the enantioselectivity of that enzyme. Okay, so those are the original sort of embodiments of this discovery. Now, I wanted to give you a more recent example, and I'm gonna go really quickly through this because I want to get to some of our stuff. This is a very important drug, sitagliptin, that earned Merck, a few $1 billion in 2015. And the original process used a rhodium catalyst. It comes back to that same hydrogenation I was telling you before. Here's the drug over here. You see it has a chiral amine. Now the precursor has an achiral ketone. And the key step that they did to get that chirality here was a hydrogenation, the same type of hydrogenation I told you about before, right? So that's what they did here. But that's very expensive to do on scale, okay? So in a second process, they optimized this. They're still doing hydrogenation, but now they're doing hydrogenation of an enamine, okay, to generate this chiral amine. So it's even more similar to what I showed before. But the thing I wanted to emphasize with it was they realized there's an enzyme called a transaminase that can take this ketone and convert it directly to a chiral amine with higher selectivity and higher activity than any of these metal catalyzed processes. I don't have time to go into the strategy they used, but I will say it took a great deal of mutagenesis in the active site, those residues around the substrate and beyond to make this work. But you're looking at a real chemical process. If you know people that take this drug, it's made with an enzyme, okay? on the world's production is made this way. And this enzyme was evolved in roughly a year to replace years of work on all these other processes. Okay, this is an example. I showed the initial embodiments. Here's a relatively recent example that's making a huge impact. Okay. Now, I wanted to tell you a little bit about what we do in my lab with this technology. One of the examples we work on, one of the enzymes we work on is a halogenase. So what a halogenase does is, as its name implies, it replaces CH bonds with C halogen bonds. The original enzyme we started working with was called RebH. It's involved in the biosynthesis of this molecule rebecamycin, which is an anti-tumor compound. The way this works, I also like this enzyme for this reason, because if you've taken organic chemistry, you know this chemistry, okay? There's a little bit of weirdness at first. It's another Flavin enzyme. That works by reacting with oxygen to form a flavin peroxide. That can react with halide to form hypohalous acid. You know this, it's just bleach, but protonated, okay? And you know that bleach is an oxidant. The weird thing that this enzyme does, though, is it doesn't release bleach into solution. It keeps the bleach within its active site, and it shuttles it to a residue, a lysine residue, that activates it so that it's an electrophilic halogen source. And what happens with an electrophilic halogen source and an electron-rich aromatic ring? You get electrophilic aromatic substitution, okay? But the trick with the enzyme active site is we have all this constellation of residues that orients the substrate relative to a key active site lysine, that lysine that activates the bleach, all right? Those are your hands that are controlling where you're going to stick that halogen atom on the aromatic ring. The mechanism is electrophilic aromatic substitution, but the selectivity is for molecular recognition, okay? So what we showed early on is that you can get the enzyme to accept different substrates, and that was neat to see. But what we really wanted to show was, could we change the selectivity? In other words, could you change how the enzyme is grabbing the substrate to force it to react at different sites? So this is important because indoles are very important compounds, and people are very interested in functionalizing them at different sites. But the way that we normally do that is to install a directing group at the one site that you can easily react with, the nitrogen. And you use that directing group to install functional groups elsewhere. And then you remove the directing group to get the compound you wanted. Obviously, what you would much rather do is go straight from the unprotected compound to the compound you wanted, leaving out the directing group installation and removal steps, okay? So we thought we could do this. This gets a little tricky. We thought we could do this by installing a deuterium on our substrate. Why did we think this would help? Well, then the wild type enzyme replaces that deuterium with chlorine, right? So you get a mass of 195. But if you make a mutant enzyme that sticks chlorine anywhere else on that substrate, you get a substrate or product with mass of 196. So that means we could run our reactions in multi-well plates. We could screen them by MALDI. This is a mass spectrometry technique. And we could look for the ratio of 196 to 195 to tell us which enzymes have altered selectivity. So I won't belabor the point, but we introduced a series of mutations via point mutations, random mutagenesis methods, site saturation mutagenesis. If we have time, we can talk about the molecular biology behind it later. But just understand that we're sticking mutations at different points in our enzyme. And we're gradually evolving it from a 7 halogenase all the way to a 6 halogenase or a 5 halogenase. And thus the selectivities are very high. They're over 90% in each case. This is critical because this means if we have a halogenase, we can switch the selectivity to other sites, okay, based on molecular recognition. I don't have time to go through the process through which this occurred in terms of activity or the effects of mutations early and later in the lineage. But if there are questions about those details, I can go over it at the end, okay? I also don't want to spend a lot of time on it, but one of the things we do in my group is after you do all that evolution, what do you do with the enzymes? We do a few different things. One is we want to try and understand how on earth did our mutations change selectivity? So we do things like solve the crystal structures. These are the crystal structures of the enzymes we evolved. We often collaborate with different groups to do things like MD simulations, molecular dynamics simulations. This tells you how residues and substrates and products jiggle around in the active site. We do that because we want to create models to predict if we have a given set of substrates can we predict where they're going to be halogenated? And this was an interesting example where we could do that. So again, you evolve the enzyme. What do? Sometimes you want to study it to understand. Sometimes you want to develop models to predict. Another thing that we very much want to do is engineer those enzymes for different synthetic applications. Remember I said at the very beginning, I like synthetic chemistry. I'm a synthetic chemist. So we've done more evolution to stabilize the enzyme, to make it work at higher temperatures with higher activity. We've evolved it to work on very large biologically active substrates. Okay, so not just change the site selectivity, but change the substrate scope. We've evolved variants that can do enantioselective halogenation via desymmetrization reactions or atroposelective halogenation. Again, happy to talk about those types of applications later. We've also used genome mining, where instead of evolving one gene, we go into sequence databases and think about ways that we can use bioinformatics tools to identify sequences that we think would be good for halogenation, and then do a lot of experimental work to confirm that. And suddenly you have enzymes that are working on very complex substrates with high activity. So another interesting case study there. So like I said, with that, you can start to think about, this idea, I think, very generally. I showed you how we started with one enzyme, RebH, that's involved in this biosynthetic pathway. And then I explained how through a combination of directed evolution, where we make mutations iteratively, right? Or genome mining, I didn't really go into it, but where we identify sequences and then you can iterate through this too. We iteratively screen sequences we identify new sequences, screen those. From all of these types of efforts, we can identify new enzymes, new halogenases that allow for synthesis of all sorts of materials. And again, we're looking at chlorination, bromination, iodination. You might notice here there's a little trick. I didn't talk about it, but we can not only halogenate aromatic CH bonds, we can halogenate olefins and do halocyclization reactions also with these enzymes. So just a broad range of chemistry, all enabled by the same electrophilic chemistry that was used in the biosynthesis of this compound. I wanted to thank the group of folks that did this. I talked about one project. This is the Flavin-dependent halogenase project here. I have several projects in my group involving B12-dependent enzymes, artificial metalloenzymes, iron-dependent halogenases, polymer degradation. We're very collaborative. We work with a lot of different groups that you might have heard of. I'm very grateful to Indiana for supporting this, and to various foundations and funding agencies for their help. We have a lot of different folks that have helped over the years. And I wanted to take a minute to just sort of mention a little bit about IU. I think it's an amazing department where we've been able to do this research. I think we have a great program. The stipend, which everybody cares about, has just been bumped up to $30K a year. In Bloomington, that goes a long way. Okay. Deadline's December 1st if you're interested, so there's plenty of time. You got to get, the normal things together. Like a lot of places, we're no longer requiring the GRE. If you have a minute to pull up the link, you can see more there. There's fee waivers available, and I'd ask you to get those in fairly soon if you're interested in that. I think we have an amazing faculty. We cover all areas of chemistry, from analytical, chemical biology, inorganic materials, organic and physical. Lots of different sort of cross-disciplinary areas if you're interested in that sort of thing. Great facilities. A lot of choices for people to work with, right? And of course, the critical thing that makes all this work are students and the resources you have to do your best work, not just in the lab, but beyond, right? So we have research centers that are funded by the NIH for chemical biology, by the NSF for materials research, for quantum science and information, and for analytical chemistry. These are great because you're interacting with a much larger network of people inherently in your research. I've been continually impressed with how students get together and form different groups, from women in organic chemistry to Nobishe to women in science, pride in chemistry. These are just their sort of somewhat recent folders for getting together. A couple have their little, they all have the little thingies. I don't know if you can see those in these slides, but just great groups of students getting together and doing stuff, right, beyond the lab, very involved with outreach and all of these things. And again, we have the more career development aspects of this sort of thing too. So great opportunities for involvement and broadening the impact of your outreach. So with that, I'm going to stop and then I guess take any questions that folks might have.
Thank you so much. I actually had a question that I wanted to ask. What are the sources for the DNA plasmids that you obtain? Where do you get those?
That's an awesome question. When I started, it's funny, when I started, actually, I'm going to go back. When I started, the universal source that everybody used was there were two. You had two options. One would be the biochemical literature. You would read something, you would find that a group had worked on something, and you would write them, and they would mail you the plasmid. That sounds pretty old-fashioned, right? Because it definitely is. If you were trying to find something new, you would literally have to go to the organism that had this enzyme that somehow you knew about and use PCR, polymerase chain reaction, to clone out the gene and then clone it into a plasmid. Okay. We don't do any of that anymore. I mean, so one of the remarkable things that's happened, again, I was a postdoc in 2000, up to 2010, and gene synthesis was still pretty expensive, right? It has just gone down so much in price that now you simply email a company, right? Or you fill out their order form and you say, I want this gene. And you can get the gene in a plasmid, and you just order it and it shows up. Now, it turns out they double the cost if it's in a plasmid. So we just order the gene in my group, and my students still clone it into plasmids. But now we use Gibson assembly to do that in one step. So you have a few options. Again, if somebody has worked on it recently, you can get it from them. If you can find the organism, you can clone it yourself. There are commercial plasmid databases like AdGene where some people will deposit their plasmids, but it's so easy and relatively low cost to order these things that nowadays that's the way we do. I would say 90% of the time. That's a great question.
Do you ever find that what's the difference in efficiency between the evolved, enzyme versus the traditional, like, real world example? Does that make sense?
So
Versus
So like if you take have you seen a difference in efficiency from the ones that you're creating, evolving, growing versus the traditional original first enzyme you started with?
Yeah. Oh, so the sitagliptin, that when I showed that Merck, that $4 billion drug, the original enzyme actually had zero activity. So how do you do that, right? This is actually one of the big problems in directed evolution. Let me show you something. I skipped over it because it's subtle. But so that's a great question. It's always good when people catch the thing you skipped over, right? So here's the thing that you want to have activity on, right? This is the precursor. It's right here, compound 11. We want activity on that. Merck was unable to identify a transaminase that had activity on this compound. So what they did was they said, well, most transaminases tend to like substrates that look kind of like this, something big and something little. And they looked at their compound and they're like, well, that's no good because we got something big and also something big. So the enzyme's just not ready for that. Okay. So they said, let's give it what it wants. Let's give it something big over here, and let's cut this aromatic compound off, and let's start screening on this compound. You guys can see me pointing at that, right? Okay. Turns out they could find enzymes that had activity on that compound. So I'm kind of lying a little bit, right? It had zero activity on their target compound, but it did have activity on this. And one of the things that's really fascinating about enzymes, so they evolve the enzyme on this compound, and it gets really good on that. And then it turns out that they put this arene back. They gave it the original substrate, and bam, the evolved enzymes now had just a little bit of activity on that compound. And now they can start evolving on that. Okay. This is called substrate walking. So you gradually adapt the substrate to what you want. You start with what the enzyme will do, and you gradually change the substrate to what you actually want. Very, very clever approach. I've used this. I used it when I was a postdoc, well before this example. So it was very common in the field, but this one made a bit more money than I did as a postdoc. So wonderful question. Now, more generally, the enzyme improvement it's all over the place, right? It's really about what is your application and how much do you need it to be improved. Nowadays, people evolve to the process. What I mean by that is if you need to make a molecule, you typically have some existing way to make it on the market. So you have economic parameters in place that tell you how good does my enzyme need to be to beat the existing, competition. And you evolve until you get that. And the remarkable thing is there are enough case studies where people have just done that. So it's a very powerful technology, right?
Mhm.
Yep.
Question?
I was more curious about what you mentioned about talking about some applications for halogenases. You mentioned that we would go a little deeper into that.
Tailed off at the end there. Applications for halogenases, but what?
That was mentioned. Just understand what exactly they're used.
Oh, yeah. So why would you evolve? Great question. Yeah. So it turns out if we go back to the original uh, inspiration here, let's start with why is this natural product halogenated, right? Well, it turns out that those chlorine substituents are massively important for its activity. Okay, so halogens dramatically affect, even though it doesn't seem like they seem somewhat innocuous, but they greatly change biological activity. So there's a huge interest in being able to site selectively install halogens, just as you see in this natural product, but in synthetic compounds, like the ones that I showed further up here, um, particularly in these types of molecules like this, because you can greatly change the potency and the biological activity more broadly. So pharmacodynamics, all the different ways that dynamics and kinetics that these things sort of interact with biological systems. Of course, the other reason is they're synthetic handles. So there's a whole family of reactions called cross-coupling, where once you install a halogen substituent, you can use that to form carbon nitrogen, carbon carbon, carbon oxygen, carbon sulfur, almost anything. Okay. Actually, I did that here. So here's a carbon chlorine bond. We actually then reacted that with an alkyne in a Larock type cyclization to generate an indole. So that's we started with no halogen. Product has no halogen, but the halogen was uniquely enabling to allow us to form this carbon-carbon bond. Okay, so I would say those two reasons. You can change the properties of your molecule directly, particularly biological properties with halogen substituents, and you also impart functional handles to do additional synthetic manipulations. That's a really good point, and that's a slide that was deleted when I fixed things this morning. Yeah, I always talk about that, but I've missed it.
We have a follow-up question?
Yeah, I was going to ask further. You mentioned pharmacological applications. Does halogenation not negatively impact humans?
Oh, no, there's not necessarily. I mean, any substitution on a molecule can be positive or negative, right? That's just the way it works. There's nothing inherently negative about halogen substitution, right? Thyroid hormone is an iodinated molecule, okay? Um, I know, for example, fluorine is in the news a lot. That's sort of a very different, it's in the news because of the types of fluorinated molecules that you're dealing with, right? These polyfluorinated compounds that are incredibly hard to degrade. You know, that's sort of a different beast. And yeah, I don't know of any example where those are beneficial, but when you're talking about a single halogen substituent or even several in these types of compounds, there are enzymes that can degrade every compound I've shown, okay? Just as there are halogenases, there are dehalogenases. It just so happens those can't deal with those polyfluorinated
Fluorinated, yeah.
hydrocarbons. Okay.
But on the other hand, there is a lot of fluorinated biomolecules, fluorinated pharmaceuticals, particularly on aromatic rings. So that's a whole area of research too, in which substitution of fluorine on an aromatic ring in whatever position tends to also increase the potency and the effectiveness of a particular drug. So with that being said, is your group also working on specific selective fluorination?
Yeah, we get this question a lot. So, yes, but not with this enzyme class. So the reason is, where do we go here? This mechanism is incompatible with fluoride.
Yeah.
Okay. So you can't do this oxidation chemistry with fluoride, and because of that, these enzymes will never do that. I
Mhm.
am convinced we work on iron-dependent halogenases also. I am
Mhm.
convinced, based on a recent paper that we published, that fluoride certainly binds to iron in those enzymes. It's around when the key steps involving the substitution of the molecule occur. It's just slightly higher in energy to stick that fluorine on than another functional group that is necessarily present in that reaction. I am quite confident that somebody is going to figure that problem out. If we don't, I mean, we're actively working on that. That would actually be for aliphatic halogenation. Um,
Okay.
Aromatic fluorination with an enzyme, I think, would be extremely challenging based on the really unique reactivity of F plus, because you
Mm.
would almost certainly need it to be electrophilic. F
Yeah.
plus is just extremely reactive, right?
Yep.
And yeah, taming that would be, I think, not the job for an enzyme. So, it's worth throwing out at that point. Enzymes aren't going to fix everything. I think they can fix a lot, and I think they can improve a lot. But there are certain things. Ultimately, it's just chemistry, right, folks? Like, so if you're doing chemistry now, you can think about enzymes. You're in the right direction. It's very difficult, I think, sometimes to train more molecular biology people to think about enzyme catalysis at a chemical level. But I think chemists are really good going the other direction. But we're also very good at seeing the limitations, right, of a catalyst that works in water and all this sort of stuff, right?
Right. Do you have, I'm pretty sure you do, summer opportunities for undergrads? I know you talked about graduate work, but what about summer opportunities for undergrads? And what's the deadline for that?
Yeah, we do, and I'm trying to think if I can quickly pull up. We we have a couple. So we're gonna have a website soon that has a general sort of way to apply for summer. Do I have any? That's a great point that I didn't have. It's all
So
grad stuff. It's
if
all
you
grad
don't
stuff.
have it available now, you could send it to me and I could send it on
Yeah,
to,
we have a couple different mechanisms that works through.
yeah.
One thing I'll say, we do not currently have a dedicated, say, NSF REU program.
Oh, okay.
We're about to reboot that. We had one. Um, what we do have are some different specific programs, and there are criteria for those. But I can send that information. That's a
All
great
right.
point.
Sounds good. So thank you very much
Yeah,
for this presentation. I learned a lot about enzymes. I'm a synthetic chemist as well.
yeah,
So, yes. So, continued success in your research. Thank you very much for accepting our invitation.
excellent. Thanks so much for the offer. Okay.
All right. Have a
All right.
good one.
Bye. Thanks.